All right, good morning, everyone. So I'd like to start by thanking the organizers. What I'd like to do today is tell you a little bit about the work that we do in my lab, trying to understand how cells move and how do they coordinate their behaviors, specifically in the context of early heart development. Now, because time is short and I am certainly going to run out of time, I wanted to start by thanking the people who did the work. So most of what I'll show you today is work by an outstanding PhD student in the lab, Negar Balagi. And I'll also show you contributions by a postdoc, Gonjar Dempsey Tandogan, and another PhD student, Chris McFall. All right, so as I mentioned in the lab, we're interested in how cells move and how do they do that collectively. And as many of you may be aware, there are massive collective cell movements involved in the early stages of heart development. And that's illustrated in this slide here, where you can see two groups of cells, the heart fields, which move from opposite sides of the embryo and meet medially, fuse, and form the heart tube. Now, we know quite a bit about the gene networks that regulate cell fate specification and differentiation from here on, but we don't know that much about what happens before, about how these cells move and how do they move cohesively as groups. So in our lab, we're trying to address those questions, and we're doing that using the fruit fly Drosophila melanogaster as a model system. Now, of course, Drosophila is famous for being genetically tractable, but the Drosophila embryo is also transparent, which allows live imaging and allows biophysical manipulations that would be much harder in many other model systems, and that I will be showing you throughout the talk today. Now, on top of that, the early stages of heart development are conserved from fruit flies to vertebrates. And you will actually see that in this movie, where what you see are the cell nuclei of two groups of cells on the left and the right, migrating from opposite sides of the embryo. And as the movie plays, you'll notice that there are two rows of cells at the front of each those two groups. These are the cardiac progenitors, the cardioblasts. And behind come a group of support cells that we refer to as the pericardial cells. And as the cells migrate and meet their contralateral partners, they establish contacts, form a lumen, and it's the formation of these lumens along the length of the embryo that drives the formation of the heart tube. Now, in our lab, we like to film this type of processes where cells move and move collectively, and then we develop image analysis tools to quantify these movements. And we've done that also in this case. So I'll use a minute for proselytism here. So we have developed image analysis tools that allow us to automatically detect and track the cardioblast nuclei. Those tools are open source and publicly available, so you're welcome to use them. And using these tools, we can now reconstruct the trajectories of every single cardioblast as they migrate to form the heart tube. And an example of that is shown here, where color indicates time. And of course, there's a lot of information embedded within these trajectories. And we can start plotting different features of the movement of the cells. And the first thing that we plotted was the velocity of the cells. And we were actually quite surprised when we looked at velocity, and color in this case indicates different cells, because what we found is that the velocity of any of these cells hovers around 0. So it fluctuates above and below 0, suggesting that the cardioblasts do not monotonically move forward, but rather take steps forward and backward as they migrate. Now, we were very interested in this stepping motion of the cardioblasts. So to investigate a little bit more how they were doing this, we decided to look at the machinery that cells use to generate forces as they move. And one of the main components of that machinery is the molecular motor myosin 2. So what you're seeing in this movie are cardioblasts and pericardial cells expressing a fluorescent reporter for myosin. And what you'll notice as the movie plays is that myosin forms really dynamic networks in these cells, specifically in the cardioblasts, that flow between the front and the back, and back to the front and back to the back of the cells. And as they're flowing in alternating position between the front and the back of the cell, they form these waves that traverse the cells between front and back. Now, those waves are so prominent that one can actually generate chymographs along the back to front axis of any of these cells, or the trailing to leading edge axis, and you get these beautiful sinusoidal patterns that represent the myosin waves as they traverse the cardioblast from the front to the back and back to the front and back to the back. Now, are these myosin waves associated with the movement of the cardioblasts? To try to understand that, we looked at cell shape changes as cardioblasts migrate. And to do that, we expressed a fluorescent reporter of cell-cell contacts, specifically in the cardioblasts. And that allows us to reconstruct the entire shape of the cell. And when we did that, what we found is that indeed, there are very dramatic cell shape changes that the cardioblasts undergo as they move, with this periodic contraction of both the front and the back of the cell, the leading and the trailing edges. Now, are these myosin waves that I showed you before responsible for these cell shape changes? Well, to address that question, we co-expressed in the same embryos the fluorescent reporter for myosin and the same cell-cell contact marker that I showed you on the previous slide. And either from the movies or from the quants for one specific cell, what you'll notice is that when myosin is at the front of the cell, at the leading edge, that leading edge contracts and the trailing edge relaxes. Now, as myosin traverses from the front to the back of the cell, the leading edge of the cell relaxes and the front contracts. And that is repeated over and over as these myosin waves go between back and the front of the cell, suggesting that the myosin waves are actually responsible for the cell shape changes, for the alternating and periodic contraction of the leading and the trailing edges of the cells that are associated with cell movement. Now, to try to understand how this contraction and relaxation of the leading and the trailing edge of the cell may be driving cell movement, we developed a mathematical model of cardioblast migration. And to start, we made a very simple model that only included two conditions. The first one is that the volume is preserved. And the second condition is that there is alternating and periodic contraction of the leading and the trailing edge of the cardioblast. And with those two conditions alone, what we get is a cell that doesn't move. And this makes perfect sense, right? Because when the front contracts, it displaces the volume of the cell backwards. And when the back contracts, it displaces the volume of the cell forward. So the centroid of the cell may oscillate, but the cell doesn't really go anywhere. Now, the beauty of mathematical models is that you can add complexity and start adding conditions to the model until you get a behavior that reproduces your in vivo observations. And of course, those conditions that you need to add make predictions about what may be happening in vivo. So we played with the model and we added different conditions. And the only way we got this cell to move in a way similar to cardioblast in vivo was if we stiffened the trailing edge. So when we do that, when we add a stiff trailing edge to the cardioblast, that limits the backward movement or the backward displacement of volume of the cells and propels the cardioblast forward. Now, is this true in vivo? Is it true that the trailing edge of a cardioblast is stiffer than the leading edge? So to start addressing that question, first we measure the deformability of both leading and trailing edges as cardioblasts migrate. And we did that by measuring the average amplitude of contraction and expansion for the leading edge in blue and the trailing edge in red over the time of cardioblast migration. And what we found was that at any given time, the amplitude of contraction or the amplitude of deformation of the leading edge, again in blue, is significantly greater than the amplitude of deformation of the trailing edge in red, suggesting that indeed the trailing edge of cardioblasts is stiffer than the leading edge. Now, we were curious about what would be the molecular machinery responsible for this stiffening of the trailing edge. And to investigate that, we decided to look at the actin cytoskeleton because actin networks can change the material properties of cell surfaces. Now, what you have here is an embryo expressing a fluorescent reporter for actin in the heart. Anterior is to the left. And what you'll notice is that as the cardioblasts migrate, there is this supracellular actin cable that forms at the trailing edge of the cardioblasts at the interface with the pericardial cells. And you can see it on both sides, on the contralateral sides of the heart. Now, we have studied supracellular actin cables in several different contexts, and most recently in the context of tissue boundary formation. So actin cables can actually form stiff borders that separate two different cell types and prevent cell mixing. So we wonder whether the formation of this boundary between the cardioblasts and the pericardial cells was actually also introducing a mechanical asymmetry in the cardioblasts that stiffens the trailing edge of the cardioblast and propels it forward. And we speculated that if that is the case, then if we disrupt the cable, we should change the oscillatory motion of the cardioblasts. Now, we have only recently started doing that experiment, and the first approach that we've taken to disrupt the actin cable is to sever it, so to cut it at 2 points so that it loses tension. And we sever the cable using an ultraviolet laser. So experiment goes as follows. We let the cells start their migration, and we cut at 2 spots. So you can see the recoil, the relaxation of tension. The contralateral site remains intact, so we can use that as a control. And then we can measure the oscillatory motion of cardioblasts on either side and compare what happens. And perhaps watching the movie is already giving you hints about what's going on, but I'll show you the quantification so we can quantify the amplitude of the forward steps. And what we find is that when we reduce tension at the cable at the trailing edge of the cardioblast, that amplitude decreases a bit. But perhaps more importantly, when we measure the amplitude of the backward steps of the cardioblast, we find that reducing tension increases that amplitude significantly. And of course, this has significant effects or important effects in the movement of the cardioblast. So if we quantify the net forward movement per step, so how far does a cardioblast go after one of these cycles of expansion of forward and backward movement, what we find is that that movement decreases significantly when we reduce tension in the actin cable at the trailing edge, suggesting once more that this actin cable introduces a mechanical asymmetry in the system that biases the movement of the cardioblast forward. So I'm going to stop here. What I've shown you today is that cardioblasts do not move monotonically forward. Rather, they take forward and backward steps as they migrate. This migration, this form of stepping motion is associated with the assembly of myosin waves that traverse the cell from the front and the back and that drive alternating and periodic contraction of the leading and the trailing edges of the cells. And we believe that those cell shape changes actually required a mechanical asymmetry in the system, which we think is introduced by the assembly of a supracellular actin cable at the trailing edge of the cardioblast that confers directionality to the movement of the cells. So with that, I'll thank you for your attention, and hopefully there is an opportunity for questions. Thanks.